Hi, my name is Andries Hadeler and I recently finished my master's thesis here at the Jacobs University in Bremen, and I would like to introduce you to the MHC class 1 and to the MHC class 1 peptide exchange. So that's the first part of the video, and then I will summarize a recently published paper of us which actually deals with one of these methods. Major histocompatibility complex class 1 molecules are a fundamental part of the adaptive immunity. These molecules play a crucial role in the defense against viral infections and diseases like cancer, due to the presentation of a broad spectrum of self and foreign endogenous peptides to the T-cell receptor of the CD8+ cytotoxic T-cells. The MHC class 1 molecule itself is a trimeric complex composed of a heavy chain, a light chain, the beta-2M microglobulin, and an 8 to 10 amino acid long peptide, which is bound in the so-called peptide binding groove of the alpha-1, alpha-2 superdomain of the heavy chain. These trimeric MHC class 1 molecules are often used in immunology to stain and identify antigen-specific T-cells. MHC class 1 tetramers are usually chosen over monomers since they interact in a more stable fashion with the TCR of the CD8+ cytotoxic T-cells. The generation of MHC class 1 monomers as well as tetramers is very time-consuming. For the production of the monomers or tetramers, the heavy chain and light chain have to be expressed in vitro in a bacterial expression system, for example E. coli. The expressed proteins are then refolded in the presence of a synthetic peptide for a couple of days. The refolded protein is then purified by size exclusion chromatography, and these purified monomers could then be biotinolated and repurified to generate the tetramers by using fluorescence labeled streptavidin. As already mentioned, the production is time-consuming, and that's why peptide exchange methods are commonly used to produce this recombinant MHC class 1 peptide complexes. As the name suggests, these methods are used to exchange a pre-bound peptide to a new incoming peptide. Typical approaches to exchange pre-bound peptides are the use of heat, cleavage of the pre-bound peptides by UV, or using mediators like dipeptides or ethanol. The latter method mentioned is described in our recently published paper in Current Research in Immunology with the title Rapid Peptide Exchange on MHC class 1 by Small Molecules Elucidates Dynamics of Bound Peptide. To first distinguish if and which small molecules are capable of acting as catalyst to exchange pre-bound peptides to new incoming peptides, we performed a mini-screen. In this mini-screen, we tested the ability of the molecules to exchange pre-bound Na9 to an incoming FITC fluorescently labeled peptide using fluorescence anisotropy. In this experimental setup, a quicker increase of the anisotropy means a quicker exchange of the pre-bound peptide to the fluorescently labeled peptide, and a higher end value indicates that more pre-bound peptide was replaced by the incoming peptide. We used glythylmethionine as a reference for the peptide exchange, since this dipeptide is known to catalyze the exchange of these peptides. As can be seen, ethylene glycol at concentrations of 10 or 20% does not increase the exchange rate when compared to the normal exchange. But when methanol was used, the exchange of the peptides was quicker than under normal conditions, but not as quick as the GM-catalyzed exchange. When we tested ethanol, the exchange rate was similar to that of the GM-catalyzed peptide exchange. After this observation, we did a molecular dynamics simulation to get a better understanding of what could be the underlying mechanism of action. In a non-restrained as well as a distant restrained model, which mimics a partially C-terminal dissociated peptide, more ethanol molecules accumulated in the MHC class 1 peptide interface, meaning between the peptide and binding groove, than with ethanol glycol. This MD simulation, in combination with the fluorescence anisotropy experiment, where we used ethanol and dipeptide together, lead us to the conclusion that the mechanism of dipeptide and small molecule mediated exchange are based on different mechanisms. We therefore proposed the following mechanism, in which ethanol extrudes the low affinity pre-bound peptide and enables higher affinity peptides to bind. If not enough high affinity peptide is in the solution, the empty MHC class 1 molecule will denature due to the instability of the peptide-free complex. The extrusion or release of the low affinity peptide is most likely caused by the accumulation of ethanol in the interface, which leads to the breaking or weakening of hydrophobic interactions.
Hi, my name is Sebastian Springer. I'm a professor of biochemistry and cell biology, and I'm the senior author of this study. I'm very excited about the work for two reasons, actually. The first one is fundamental science. It's allowed us to show how these small molecules can break up the interactions between the class 1 molecules and the peptides. And peptide dissociation and exchange is important in the process in the cell that we call peptide optimization. So there is some fundamental importance to this. But then there's also an applied side. Of course, the work allows you to take a complex of a class 1 molecule with one peptide and turn it into a complex of the same class 1 molecule with another peptide. That can be important in screening biotechnology for all kinds of reasons. So I hope that you enjoyed the video, and I hope that you'll read our paper.